allow me to thank the IVES for the organization of the kind meeting and the possibility to speak about my research in the topic of non-Saccharomyces yeast. This is a trend topic today, probably you know, but the history of the evolution of yeast in enology is very long. I think that at the beginning of the 20th century, enologists and also scientists discovered the role of Saccharomyces in wine fermentation. And then after that started a very big, a very long work of selection of different Saccharomyces strains. And after that, the large diffusion of active dry yeast in winery around the world. This certainly ensure a big control of alcoholic fermentation and an increase of general quality of wine, but at the same time rise concern about the loss of biodiversity in enology, because only one species, Saccharomyces cerevisiae, dominate the microbiota probably in all winery of the world. So, starting from the 20th year, there is a new interest in exploring the biodiversity and in the application of non-Saccharomyces yeast in wine. This is a trend topic, but a very young topic, because if you observe this review, it is dated in 2002, the majority of enological yeast are classified as contaminant or spoilage. So the idea about this yeast is changed very recently, and some work are in actual today. This is the trend of research about this topic. This starts very fastly at the beginning of the 20th, and then grow in the last year. If you read this large biography, it's possible to find about 5 different types of application on non-Saccharomyces yeast in enology. The removal of toxic compound from grape must or from wine, particularly as example ochratoxin A, or heavy metal, or biogenic amine, for example, also. The improvement on the flavor and the taste, the biocontrol against the spoilage microorganisms, in particular against Brettanomyces and lactic bacteria, the reduction of ethanol accumulation, this is a very important topic today, and finally the regulation of acidic profile. About this, this is one of the applications they tested. The first experiment started in the 19th year, but in this period, the goal is the reduction of wine acidity. It's incredible today, but this is the goal in this year. Because in the 90s, the selected culture of lactic bacteria are not performing as today, so there is necessary to find an alternative at the lactic bacteria. And some researchers proposed the use of Schistosaccharomyces pombe as alternative to the lactic acid bacteria, because this yeast is capable to reduce the acidity consuming malic acid through the maloalcoholic fermentation. So to take malic acid, and transform it in ethanol. Today the situation is completely changed. The goal is the increasing of the wine acidity using lactate thermotolerance. It is an yeast that is able to take a little bit of glucose present in the grape must and not transform it in acetate or ethanol, but convert it in lactate, increasing the total acidity with the addition at the grape must of a stable lactic acid, stable acid. Another interesting topic is the improvement of flavor and taste of wine. Numerous studies have reported that the mixed or sequential fermentation with non-Saccharomyces and Saccharomyces produce great varieties of volatile content, because the non-Saccharomyces yeast have some enzymatic activity more expressed than in Saccharomyces, then is able to extract more aromatic precursors from grapes, for example, or better produce more fermentative aroma. I report 2 experiments that are recently published in one. The first is the biological acidification of Vino Santo. It is a traditional sweet wine produced in different regions of Italy using lactate thermotolerance, obviously. And the second one is the use of non-Saccharomyces yeast in the Prix de Mousse of Lambrusco, that is a sparkling wine produced in the north of Italy. Okay, the Vino Santo is a traditional sweet wine produced in different regions of Italy. The main production is in Tuscany, but also there is a very traditional production in Veneto, in Emilia-Romagna, and also in Trentino Alto Adige when I come. Obviously, different grapes are involved in this production. For example, Trebbiano in Tuscany, Garganega in Veneto, Malvasia in Emilia, and the Nosiola in Trentino, but also Sangiovese, because there is a little production of Vino Santo from red grape in Tuscany. In all cases, the process is more or less the same. The grapes are harvested early. There is a very long natural drying in ventilated room. This natural drying traditionally go until ester, and the crushing was performed in the only week before Easter. So this is the name Vino Santo derived probably from this tradition. After grape crushing, the mousse were put in a little barrel, which is called caratelli, and ferment very slowly in oxidative condition. The key to obtain a high quality Vino Santo is to maintain a high acidity in the grape mass to counteract the spoilage microorganism during the fermentation, because this process is very, very long, and if start, for example, lactic bacteria, probably there is the problem of stability of wine. So it's necessary to maintain this acidity, and the idea is to maintain this acidity using Lancaster thermotolerance. So I have tested, I performed 4 different trials, the normal production using only Saccharomyces cerevisiae, and 3 different trials using Lancaster thermotolerance before inoculation of Saccharomyces cerevisiae. As usual, in this case, Lancaster thermotolerance was inoculated 5 days before Saccharomyces cerevisiae. Another point is the nutrition, the nitrogen supplementation. I have tested 2 typical nitrogen supplementation, one using inorganic nitrogen and one using organic nitrogen at the level usually utilized in the winery, and another test with the double nutrition, so a very strong nutrition with organic nitrogen. So this is the trend of fermentation. I hope to see, because it is too little, but you observe the blue line is the fermentation performed by Saccharomyces cerevisiae. The only experiment using Lancaster which is able to have the same performance is the experiment performed with the double organic nutrition, if you observe. The other 2 have a less activity of fermentation and consume less sugar than the other. In this case, it's not a very big problem, because it's a sweet wine, so a residual sugar is expected. But this is an indication that the presence of mixed yeast culture increase probably the nitrogen requirement, and probably cause the problem at the activity, or in more cases, the alteration of the activity of Saccharomyces. The same thing is possible to observe the fermentation rate, the evolution of the fermentation rate. Obviously, in the mixed fermentation, there are a lot of cells, so the fermentation rate in Italy is higher, but the best result is obtained also by Saccharomyces cerevisiae. So this result could be probably related at the yeast stress for the cohabitation in the same environment of these 2 species. Regarding the acidity, this is the focus for the use of Lancaster. In effect, the use of Lancaster ensure the reduction of pH during the alcoholic fermentation below the critical interval between 3.5 and 3.3. That is a typical interval for activity of spoilage lactic bacteria. But if you observe them, the best performance is obtained with the mineral nutrition, which is not the best fermenting Lancaster thermotolerance. So it's not easy to find a balance between the fermentation performance, which is fundamental, obviously, and the secondary metabolism of the other yeast. Also, the characterization of the wine, the chemical characterization of different wine, confirm that this lowering of pH is due to the production of lactic acid, which is higher in the test performance, but Lancaster thermotolerance and nutritional mineral nitrogen nutrition. But if you observe, there is a big difference in the profile of volatile compounds, obviously between the test performed with lactate thermotolerance and the test performed only with Saccharomyces cerevisiae, but also between the different tests performed on lactate thermotolerance. So the nutritional protocol is a key point in the regulation of activity of non-Saccharomyces wine, not only in terms of trend of fermentation, but also in terms of expression of secondary metabolism that give the wine distinctive characters. The second experiment is performed in the Lambrusco region. Lambrusco is one of the major oenological production in Italy. Typically is a wine that is not considered very high quality, and typically is also a cheap wine, but is firmly among the three most exported Italian wine by volume and also by value. In particular from the USA and from the north of Italy. It's a very particular wine because it's a red, sparkling, dry or semi-sweet wine consumed in the early months after bottling with an organic profile based on the note of red fruit, a good acidity, a moderate tannins, and an ethanol contents no more than 11%. To obtain this Lambrusco style, the winemaking is based on the Charmat method, with a one-step alcoholic fermentation in a pressurized tank in autoclave and an early isobaric bottling. This is today the most widespread production process, but traditionally Lambrusco wine are made by Champenoise method. So to improve the quality of the Lambrusco produced by Champenoise method, I tested two different strains of non-Saccharomyces yeast, Anseniaspora Guillermondi and Torulaspora del Brechi, in mixed fermentation with Saccharomyces cerevisiae during the battle fermentation, so during the second fermentation. This is the particularity of this work because typically, generally it's considered that the non-Saccharomyces yeast are not resistant at ethanol. To produce sparkling wine with obviously distinctive character. So the case is the adaptation of these two non-Saccharomyces wine, Saccharomyces yeast, sorry, at the ethanol content of wine. So I performed three different pieds de cuve that last 48 hours, increasing gradually the amount of wine in the mixture to adapt yeast at the ethanol and obtain a good result because obviously in the case of Saccharomyces cerevisiae obtain the higher cellular concentration, but this is obvious. But at the same, the concentration that I obtained in the case of Anseniaspora in the same order of magnitude, and also more or less in the case of Torulaspora del Brechi. So I performed three tests, one with pure Saccharomyces cerevisiae, one mixing 1-to-1 ratio the Saccharomyces cerevisiae and Torulaspora del Brechi, and the last one mixing 1-to-1 Saccharomyces cerevisiae and Anseniaspora Guillermondi. And then they put the wine in bottle fermentation with an yeast inoculum that is about 1 million of cell for milliliter. This is a classical inoculum to make a sparkling wine. So this is the fermentation trend in bottle. The blue line is also Saccharomyces that has best performance, this is obviously. But if you observe, after 10 days, the trend of fermentation of the other two tests are more or less similar, and the consumption of sugar are concluded at the same time. So, So I obtained a good result in all tests. This is the fermentation rate. It is obviously higher in the case of Saccharomyces cerevisiae at the beginning of the test, but after 10 days, also in this case, there is not statistically different. This is the cellular concentration. In the test with Saccharomyces, obviously there is only Saccharomyces. In the other case, the concentration of non-Saccharomyces yeast remain higher for a long time. So this is demonstrated that adaptation of non-Saccharomyces is good, and the ethanol resistance is very interesting. Also in the battle condition, that is very very harsh condition for yeast. I performed also the chemical characterization of volatile profile. So the blue dot is the basic wine, which is obviously very different from the wine after bottle fermentation, obviously. But you observe there is a strong difference between the wine fermented with Saccharomyces cerevisiae and the other two in the main classes of aromatic compound. So in conclusion, the non-Saccharomyces yeast are an interesting tool in winemaking. The selected culture of this yeast are viable today because all these experiments are performed with commercial culture, not with experimental strain. So is data easily replicable in the winery. The selected culture of this yeast guarantee the promised result is effective. An adequate acclimatization of non-Saccharomyces yeast can extend their field of use even in wine with high limiting factors such as ethanol. Ethanol tolerance appears to be greater than expected or than published in the previous work. The addition of an high concentrated non-Saccharomyces yeast in winemaking, in addition to Saccharomyces, seem change the microbial ecosystem and induce effects which are not easily predictable also in Saccharomyces cerevisiae. This I think is the key point today because there is no more data about the use of non-Saccharomyces yeast in combination with Saccharomyces. So it's very important to validate its use in each winery and in each practical condition. So a careful validation of this yeast is essential, pay particular attention to the nitrogen nutrition, which is today is a key aspect also for the climate change, which change the composition on grape. Thank you.